It seems to me that there is a particular responsibility for those of us who have been involved in the kind of engineering that has turned into synthetic biology to get it right, because there is an issue of public confidence in what we're collectively trying to do, and I think that up to now, as a community, we've been very careful, and rightly so, to make sure that what we do is transparent and assented to by people who are not necessarily scientists or specialists. And this is true of the idea of using synthetic biology or pathway engineering, or whatever you want to call it, to engineer the production of new drugs. And so what I'm going to talk about today has, in part, long roots in my personal research history. It, I hope, will tell you some new information, which I'm very excited about in this area. And I want to finish with what I think is a major challenge that we can now address because of the tools of synthetic biology. I don't say it's done, I just think it's possible. And I'm delighted I'm not the first in this meeting to put up Charles Darwin. Of course, we've been celebrating him, particularly in Cambridge in the last couple of years. And one of the first things that I want to remind you of is that natural products, that is, the small molecules that you can consider as nature's chemistry set, are special because they are shaped and preserved by evolution. That is to say that they are genetically encoded and subject to all the pressures that every other constituent of life forms is subject to. And on the next page, I'm not showing you this for the iconic first picture ever of a phylogenetic tree, and I'm not going to ask you to read his scrawl here, but I'm showing you this because of that scribble in the margin, because he realized many years before he published, we don't have that luxury now, he realized that his theory implied that extinction had to happen. And so I want you to think about the evolution of natural products. I'm now converted to the idea that we shouldn't look for a billet for every bullet. We shouldn't look for a specific function for every natural product. Rather, it seems to me that Vernon Jones's idea that the trait that is evolvable and confers an advantage is the ability to produce such molecules. It's a sort of shadow, a foreshadowing, if you like, of the vertebrate immune system. We don't demand that every immunoglobulin has a specific role. And part of what I'm interested in is the natural evolution of these systems. But along with that goes the wish and the drive to engineer them ourselves. And those things go hand in hand. So I see every natural product as a representative of a virtual library of similar compounds that we could, because they're perfectly stable and sensible chemicals, could be recreated by us. And those of you who don't know what a polyketide is, you might at least see that it's a polymer, might well recognize the individual members of this large tribe of chemicals. We've already heard about Rapamycin as one of the 4 components of a now 40, 50-year-old therapy against TB. Rapamycin is a soil bacterium of the Easter Island produces this, and I'm going to come back to that. Epithyllone has featured already in a previous talk. Amphotericin, which is a ghastly drug that crystallizes out in your major organs, is still the drug of choice in UK hospitals for life-threatening fungal diseases. It's given in the form of liposomal preparations to try to make it slightly less unbearable. And in the middle is the molecule that I first got interested in. And if you know where to start at about 8 o'clock, you can follow the chain round. And if you strip off the sugars and you think about it, you can see that this is a polymer of one starter unit, basically a propionic acid, very, very simple acid, and 6 further 3-carbon units. So the number 6 is important because it's like a fatty acid biosynthetic enzyme. It knows how to count, and it does different things at each stage. And so let's just dwell for a moment on this historical finding. Much to our surprise, and at about the same time, Leonard Katz's group in Abbott Laboratories made exactly the same finding. It turned out that instead of using the same set of enzymes to build up a chain, which would have been very sensible and very economical, and to have the changing shape of the substrate itself dictate that different things happened, that was my original idea. What actually happens is that you use a different set of enzymes, very similar to the previous set, to add the next unit, and then you discard it. Or to put it another way, you actually move the growing chain through space along this track, exactly a molecular equivalent of a car assembly line. And what you see is what you get if you have more enzymes, and each of these circles represents an activity, then more chemistry happens. So you have full reduction here at module 4, whereas here you keep the keto group. If you kept all the keto groups, it would be a polyketide, and that's how they get their name. But actually what happens is that you get a mixture of things, a mixture of stereochemistries, and the number 6 is the number of modules, so of course you only add 6 and then it stops. And this immediately explained why hundreds, if not thousands, of similar complex chemicals in nature all are derived from a single simple pathway, by just modulating the detailed composition of the modules. So this looks like a type of enzymology where you have to redefine what it means to be a selective catalyst, because you're defined by where you are, and your substrate is presented to you. You're not given the universe of cellular components as potential substrates. You're given, in general, a single substrate. And what was immediately realized was that this meant that you could imagine tinkering with it. And so in the next few minutes, I'm going to talk about that promise of natural modularity. And you can see how this would fit with what we've talked about, and the problems with it. And then I'm going to encourage you by saying that the spin-out company, Biota, has, despite those problems, managed to successfully create a focused library of bioactive polyketides that are going to be useful. And then I'm going to set a challenge. This is work that hasn't been done yet, but which I think synthetic biology makes possible. So here's the promise. This experiment was actually done. You rub out an enzyme that normally reduces a double bond. Everything else works, and you get an analog with a double bond which wasn't there before. And this is another very early experiment. Now that you've sequenced 2 different clusters, you can use one as spare parts that dictates a unit without a methyl branch. And when you substitute that in a little model synthase that makes a small ring compound instead of a macrocycle, a methyl group disappears, which, if you're trained as a chemist, as I was, has a certain charm, because a bacterium's doing it, and it can do it kilograms at a time in excellent yield. And that technology, called domain swapping, has been a mainstay of this field for 15 years. And indeed, people have thought and tried almost all the strategies you could possibly imagine, whether it's changing the nature of the extender units or the starter units or the oxidation states or the stereochemistry or the ring size, or indeed the sugars. I'm not going to talk too much about it. So it does work, and here's a real picture from a paper, not by us, with this sort of wallpaper design of about 50 new analogues. The snag is, if you read the small print, that they're not produced very efficiently. In fact, they're produced on alternate Thursdays in a mass spectrometer, if you're lucky. I rashly, to Leonard Katz, call this homeopathic quantities, and he said in his best New York accent, Peter says this is homeopathic, I call it homeopathetic. And he's right. So the reality is low yields. In fact, maybe wrong products, extra products, but very often nothing at all. And why aren't they modular? Well, it may be that the chemical that's attached, because remember the intermediates are not released, is simply not liked by the gateway ketosynthase of the next module, because this is the key enzyme. This is the one that's going to recognize the carrier and the growing chain on it, and is going to accept the elongating chain and do something to it. So if the ketosynthase says no, that's game over. And it could be that it doesn't like squares, it'll only have circles. Or it could be that the docking onto the acyl carrier protein is wrong because you've given it a different acyl carrier protein and it doesn't like it, even though it's a circle that's attached. Or it could be that the tether is not a blind tether, but actually an important piece of structure. And so, can we do better? Well, yes, we can do better in the ways that we heard about from previous speakers, that you can build up your databases and your methods of doing things. You can make synthetic genes, and people are doing that. But basically, we need structural information and a better knowledge base. And just to show that diagrammatically, there is now some, at least partial, X-ray crystal structure. Instead of a picture of each enzyme being loosely tethered in a very relaxed way, we now think there's very little wasted structure. So we can be more careful about the way we cut and paste, and that does mean we do better than we did before. And if you go down to the level of single amino acids, we know from work in Cambridge by my colleagues Kira Weissman and Bill Broadhurst that the reason that DEBS3 doesn't get out of turn and start docking on the end of DEBS1, and DEBS2 instead always docks on DEBS1, is because of an exquisite alpha helical handshake, in which, when you really look down at it, there is an electrostatic positive interaction when you have the right partner, and take my word for it, that isn't there when it's the wrong partner. And so what we learned in Cambridge and transferred to Biotica is this idea that this bit in here is quite tough. We can do some of it. But what we need to build up our number of molecules is to learn how to vary the front part, so the building blocks are different going in, and to build up the so-called post-polyketide synthase part, because then you start to multiply the numbers up. And here's Biotica's biography, and I don't want to take you through every word. We started it in 1996. We made some mistakes. We made a profit, actually. We paid taxes. That was a bad mistake, which we soon corrected. And then in 2000, we got a commercial contract from the business unit at Dow Agro. They were not sure whether to be pleased or startled when, after 18 months, we'd broken every one of their core patents on a molecule which for them was extremely important, a very nice compound called spinosad, which won the mother company, Dow Chemical, the President's Green Chemistry Award, which they'd never won before and probably won't again. And in 2002, we got funding, and I won't take you through all this, but let's just say our best, most forward candidates are now in advanced preclinical studies. So what are they doing? Well, the successful example that I'm going to quickly go through is rapamycins. Here it is, 14 cycles of polyketide chain extension and an amino acid. You don't need to know about the biology. It's perfectly sensible. The protein target of rapamycin should be called the mammalian target of rapamycin. And it's absolutely key to the division of cells, for example, the cells of the immune system. So originally, rapamycin was an antifungal and then an immunosuppressant. This is a brutal summary of the fact that after several years, and not many millions, frankly, of money, they have 120 plus novel rapalogs in good yield by fermentation of the original host, which has been improved to meet the demand. And here's just one where you take away this methoxy group and you get something called BC210. You can feed to this so-called RAPK strain novel starter acids, and you do something called MUTA Synthesis, and you can feed the whole catalog if you want. Many go through, and just one piece of biological data. It simply says one BC210 partitions across the blood-brain barrier much more favorably than the parent molecule. And as an anti-cancer against a brain glioma, that would be good. And here's just a bit of fun. An experiment where nearly 2,000 mice were allowed to become middle-aged because they couldn't get the drug into the mice, and they discovered, nevertheless, they got a Nature paper out of it because the lifespan was increased for all these mice by an appreciable amount. And it hit papers all around the world, the Belgians in particular, elixir de jouvence, they had no doubt about it at all. And the exciting thing I want to give you 2 slides on is that antivirals are tough to find. And just recently, they have recycled a drug thrown away by Novartis 15 years ago because it was insoluble and it didn't fit the rules of 5 and all that sort of thing. And what this drug does, it binds to a host function that a virus needs, and that means the barrier for overcoming that drug is much harder. And they have good evidence that this compound called sangliferin, which was developed in combination with a lab in China, in Shanghai, the Chinese Academy of Science, and licensed to Biota. And it's a totally different structure to the clinically tested cyclosporins. And we think it's got a lot of charm, and we've generated first and now second stage precursors. We think that in here there is a real opportunity for antiviral drug development. And this was really done on, I mean, we're not just lean and mean in Biotica, we're positively anorexic. And yet we can produce these things. So here is the challenge for the future. Even if you can tune and know about structure, an unsolved problem is that if a ketosynthase doesn't like what you're giving it, you will never have a really combinatorial production of new things. And so what we did was we tried to think if we could learn from nature. And while we were pondering that, some work that we did with Institut Pasteur scientists led by Stuart Cole and Caroline Demangel showed us that we actually had in our hands something that would fit the bill. Unfortunately, it had some problems. This is what happens when that polyketide is administered to a small human by the vector. We think it's a mosquito, so Luke, maybe we should talk. And Buruli ulcer is actually more prevalent in West Africa than leprosy, and nearly as much as TB. And there's no drug therapy to speak of, no vaccine, you're disfigured for life. It's absolutely painless and there's no immune response. And the theory is that this is made by an assembly line enzyme too. This side chain as one assembly attached then to this core. And on the plane over to Paris, I wrote down the gene structure. And after a year, we saw the gene structure, and here it is. It's the biggest polyketide synthase and the most brutal that I ever had to try and sequence. And the reason is the internal homology makes it look as though it's evolved last week. There are 18,000 amino acids in this polypeptide chain. This is just a bacterium, and it's got 18,000 amino acids. And you can see I've highlighted the ketosynthases, and there are lots of them. All 16 are sequence identical, even though the substrates that they handle differ. And so it looks as though if you were to shut your eyes and change the position of any of those ketosynthases, it would still work. So maybe this is the combinatorial set that we've been looking for. But the trouble is, the biologists among you will know, if you put 16 sequences that are identical in tandem array, it will collapse. And so what we need is synthetic biology to synthesize these modules, varying the DNA sequence but keeping the protein the same. And that's what we intend to do. So that's a work in progress. And thank you for listening.